Hi, good afternoon. My name is Miao Xu. So this topic is about the megakaryocyte development and also the platelet formation. Megakaryocytes, like all the other cells in the blood, develop from a master stem cell. So committed megakaryocyte precursor cells develop from potential hematopoietic progenitors expressing surface CD34 and CD41. From cell-filled germ, they have biopotential to develop into both megakaryocytic and erythroid cells. Here, we will focus on the megakaryocytic lineage. The pro-megakaryoblast is the first morphological recognizable megakaryocyte precursor in bone marrow. Megakaryoplasts are 15 to 50 micrometer in diameter and large all nuclei and basophilic cytoplasma lacking granules. The megakaryoblast or stage 1 megakaryocytes is a more mature cell that has a kidney-shaped nucleus with 2 sets of chromosomes. The pro-megakaryocyte or stage 2 megakaryocyte is a 20 to 80 micrometer in diameter with a polychromatic cortex. The cytoplasma is less basophilic and now contains developing granules. And then the development reaches the first milestone, endomitosis. It becomes polyploid through repeated cycles of DNA replication without cell division. And polyploidization is essential for efficient platelet production. The left picture here shows multiple mitotic spindle pore formation during the endomitotic process of thrombin treated megakaryocytes. Although the number of endomitotic cycles can range from 2 to 6, the majority of megakaryocytes undergo 3 endomitotic cycles to attain a DNA content of 16N. After the process of endomitosis is completed, the megakaryocyte begins a maturation stage in which the cytoplasma rapidly fills with platelet-specific proteins, organelles, and membrane systems that will ultimately be subdivided and packaged into platelets. The maturation involves several processes. Here, we show the demarcation membrane system, dense tubular system, and granules formation. One of the most striking features of a mature megakaryocyte is its elaborate DMS. It is the network of membrane channels. The suborganization of the megakaryocyte cytoplasma into membrane delineated platelet territories was first reported by Coase and Dimash. The exact function of this elaborated smooth membrane system has been hotly debated for many years. Initially, it was postulated to play a central role in platelet formation by defining preformed platelet territory within the megakaryocytoplasma. However, recent studies more strongly suggest that the DMS functions primarily as a membrane reserve for proplatelet formation and extension. Megakaryocytes contain a dense tubular system, and the dense tubular system is believed to be prostaglandin synthesized in platelets. Megakaryocyte maturation is characterized by the progressive formation and appearance of a variety of secretory granules. The most abundant alpha granules, which contain proteins essential for platelet adhesion during vascular repair, and then dense granules, or dense bodies, you may call it, identified in electron micrographs by virtue of their electron-dense cores, contain a variety of hemostatically active substances that are released upon platelet activation, including serotonin, ADP, ATP, and calcium. Although it has been universally accepted that platelets derived from megakaryocytes, the mechanism by which platelets form and release from these precursor cells remain controversial. 3 modes have been proposed to explain the mechanics of platelet production. The first is cytoplasmic fragmentation via the DMS. In this model, the DMS defines predetermined platelet-sized fields within the megakaryocyte cytoplasm. Platelets form when the cytoplasm fragments along these DMS fracture lines. The second one is platelet body. In this model, platelets pinch off from blasts protruded at the megakaryocyte periphery. The third one is platelet formation. The flow mode of platelet formation requires platelets to form through platelet intermediate structures, which are long cytoplasmic extensions that appear as platelet-sized beads linked together by thin cytoplasmic strands. In this model, the DMS functions primarily as the membrane reservoir for extension of platelets. Here, we see the picture. You may see the platelet processes from these long strands into the interior of blood sinews in the bone marrow. As megakaryocytes transition from immature cells to release platelets, a systematic series of events occurs. First, after commitment to the megakaryocyte lineage, cells undergo mitosis and transcriptional activation. And then, during cytoplasm maturation and expansion, the megakaryocyte undergoes organelle synthesis and platelet-specific protein amplification. Microtubule arrays from the centrosome are clearly established here. And then, prior to the onset of platelet formation, centrosomes disassemble and microtubules translocate to the cell cortex, followed by the platelet production initiated with the formation of large pseudopodium that elongate and form thin platelet processes with bulbous ends. These ends contain a peripheral bundle of microtubules that loops upon itself and form a teardrop-shaped structure. Sliding of overlapping microtubules drives platelet elongation as organelles are moved individually over microtubules into platelet's end, which nascent platelet assemble. And then growth and extension of the platelet processes is associated with repeated actin-dependent bending and bifurcation, which amplifies free platelet ends. Platelets form constrictions along their length, giving them a beaded appearance. And then the entire megakaryocyte cytoplasma is converted into a mass of platelets, which release from the megakaryocyte body after rapid retraction. A bubble of 2 platelet-sized particles linked by a small cytoplasmic bridge will provide an intermediate stage. Platelets undergo further fragmentation into individual platelets, and the extruded naked nucleus remaining after near complete shedding of the megakaryocyte cytoplasma undergoes apoptosis. This is all of our course, and thank you very much.